I'll be discussing our research project that we're working on with SAMRI and the University of Adelaide. Like Anne said, it's looking at blood biomarkers within plasma to differentiate between transient ischemic attacks, transient ischemic attack mimics, and minor ischemic stroke in a clinical population. This is aimed at reducing the subsequent risk of ischemic stroke, and I'll talk a little bit about that in a little bit. Just for a bit of background, we know that stroke is not only a leading cause of death, but also disability worldwide. And we know that a majority of strokes tend to be ischemic. What this means is that these strokes are caused by a disruption in blood supply to the brain. So because the brain is quite dependent on a constant supply of oxygen, as soon as there's an interruption in the blood supply, it results in very rapid neuronal cell death. And that is really why when we're managing a stroke, time is always of the essence. This can always be caused by either a blood clot that's happened within the brain and it's interrupted the blood supply, or it can be a blood clot that's formed elsewhere in the body and it's traveled within the bloodstream to then interrupt blood supply to an area of neuronal tissue within the brain. The symptoms that you see with clinical presentation for a stroke will always correlate to where a stroke has happened within the brain. So, for example, if the supply to the Broca's area in the frontal lobe is interrupted, which is responsible for speech, one of the main symptoms that you will see primarily will be issues with speech production or speech disturbances. There's quite a few risk factors involved as well. So atherosclerosis is a really main one because, again, it's that plaque buildup really exacerbates the formation of a blood clot, and then that kind of leads to an increased risk of ischemic stroke in the population. Transient ischemic attacks, which I'll refer to as a TIA for the purpose of this talk, is quite similar to an ischemic stroke in the way that they develop. So a TIA is also caused by a disruption in blood supply. The main difference here is just that the disruption is always temporary. So the symptoms that you'll see with a TIA will always resolve and usually quite rapidly. And these symptoms will also overlap with an ischemic stroke. So they can be something like dizziness or even confusion or even limb weakness, depending on where that disruption has happened. A lot of TIAs will then eventually lead to an increased risk of ischemic stroke. So a lot of patients that will come in with a TIA will go on to have an ischemic stroke. And quite often this happens within the first month following the initial onset as well. There's a few issues with diagnosing TIAs, and that's really where our research comes in as well. So because the symptoms resolve quite quickly, if a patient is presenting with something like confusion and it resolves, they're probably not likely to realize that they've had an ischemic event. They might just think it might be relatively benign and they might just chalk it up to something else. Whereas even if the symptoms are quite severe, by the time they go to a clinician and are seen clinically, those symptoms have already resolved. So there's a lot of reliance on patients' self-reported accounts of the experience. There's also, because there's no neuronal cell death and no tissue death that's visible on imaging in the same way you would see with an ischemic stroke, it's a lot harder to diagnose. And at the moment, we don't really have any sort of clear, accurate tests to diagnose a TIA as urgently as we should be able to. And this has really affected the rate of misdiagnoses that happen with TIAs. And there's been quite a lot of research on this as well. So we've looked at the accounts of ED physicians and also specialist neurologists. And the inter-observer agreement between those groups has not ideal. And there's quite often a misdiagnosis. So a TIA can get misdiagnosed as a migraine or even a seizure. Sorry, the lights in my office have just gone out, so I'll leave it as that, but I might fix it in a second. And of course, when you're misdiagnosing an event, it's then going to affect the way a patient is being treated and then managed subsequently. So if a TIA was being identified really early on in the process, you can manage and treat a patient with the aim to reduce the likelihood of an ischemic stroke in the future. Whereas if it's being missed completely, that risk is not really being managed at all. that's kind of where biomarkers come in as well, and that's what our research project focuses on. So similar to the way that troponin is used in cardiology, where the higher expression is investigated to measure cardiac risk, we are aiming to look at plasma biomarkers to try and differentiate between a TIA, a TIA mimic, so something like a migraine or an epileptic seizure, and even minor stroke, to try and identify those and measure those differences in expression really early on, so that they can be managed and used appropriately for future reduction of risk. So FASTIT, which stands for To Find a Simple Test in TIA, is based mainly at the Royal Adelaide Hospital in South Australia. We enroll patients primarily from the emergency department and the stroke unit, and that has been an ongoing project, at least for my PhD, since 2021, but we actually started before that. I do have a QR code on the screen if anyone is interested in looking at our protocol as well, and that just kind of lays out all of our methodology and our design in a lot more detail. It's funded primarily by the Hospital Research Foundation, and we have quite a large group of multidisciplinary medical scientists and specialist neurologists, and even vets who are quite involved on an ongoing basis to inform how we work on the study. The aim overall, like I've said, is to use plasma samples in a patient cohort from the Royal Adelaide Hospital to identify a panel of biomarkers across a few different modalities. And I'll talk a little bit more about that in just a couple of slides from now. So we approach patients who are presenting with TIA-like symptoms at the Royal Adelaide. So at the time that I, for example, approach a patient, I'm blinded as to what their final diagnosis is or what they are being assessed for. I am only given the symptoms that they've come in with. The reason we've done this is so that we can enroll confirmed TIAs and TIA mimics, as well as minor strokes, because there is quite a bit of overlap in the clinical presentation for those conditions. And then all these samples get classified retrospectively by a panel of neurologists later down the line. We don't enroll patients who are presenting with a hemorrhagic stroke, and we've also had patients who have had symptoms that fall within our criteria, but they've also got an active tumor, for example, just to avoid those confounding factors, and also because we're more interested in the ischemic cascade rather than an aneurysm or a hemorrhage as well. All those samples are collected within 48 hours of the onset of symptoms and are processed and stored within SAMRI until they are then analysed as a batch for the various arms of the study, which I'll talk about in just the next slide. There are a few different aims. So the primary aim is that we have been doing discovery multi-omic investigation on these plasma samples. That's included primarily proteomic analysis, and then also a combination of metabolomic and lipidomic analysis as well. We are also looking at oxidative stress and the expression of azaprostanes following those events in these patients as well. And we also collaborate with Dr. Roman Kostecki at the University of Adelaide and use an in vivo biosensor to measure those changes in the same cohort as well. In conjunction with all of this, we've also got obviously the demographic data that we've collected and also some clinical data, so imaging reporting outcomes, and we've used the modified Rankin scale to measure outcomes following 90 days following enrollment as well. And then that's combined with the classification that we received from the neurologists as well. So just focusing on proteomics. We collaborate with the mass spectrometry group at SAMRI that is focused on doing a discovery proteomic investigation using mass spectrometry as well. We were initially using a DDA passive methodology, which is data-dependent acquisition, and that has since been updated following quite a lot of method development to a DIA passive methodology, which is data-independent. What that's done is basically allow us to measure all the protein IDs that we can, regardless of abundance and expression levels, and just expand for more sensitivity and specificity in our cohort as well. Similarly, we've done discovery lipidomic analysis, also using mass spectrometry, and also done a pilot for a metabolomic investigation as well. So that has helped us kind of compare levels of, aspirin, saccharin as well in our patient cohort. And we've found some pathways that are kind of in line with what you would expect to see in a stroke cohort primarily. So, proteins that are involved in inflammation or lipids that are involved in atherosclerosis, finding those being expressed in our cohort of patients has kind of validated what we were expecting to see. But this is an ongoing analysis, and this is still an ongoing expansion of our initial pilot as well. Just going on to oxidative stress, we know it's a key component of the ischemic cascade as well. And isoprostanes are quite often used as a marker of oxidative stress, and protein carbonyls have also been investigated as a marker, specifically in acute ischemic stroke as well, and as a predictor of stroke severity. So we collaborate with the Women's and Children's Hospital and SA Health in South Australia to do a candidate lipid analysis, looking at isoprostane expression following the onset of these TIAs or TIA mimics or minor strokes, and that also uses mass spectrometry. Like I've mentioned, we collaborate with Dr. Kostecki at the University of Adelaide, and he has developed an in vivo biosensor that has allowed us to measure the protein carbonyls in the same cohort as well to try and validate those as a combination. And again, this is an ongoing analysis as well. In addition to all of this, we've also got reporting outcomes from the patients that we've enrolled and kind of whether they had an MRI or a CT and what those outcomes say, and kind of comparing those with how the patients have been classified, whether there's consensus in that classification, combined with the demographic data as well as the clinical data that we've got. I think that kind of develops a very well-rounded study because we're measuring all these different variables to try and develop a panel that's multivariable and multidisciplinary as well. In terms of future direction, so we've got reporting outcomes for imaging, but I think it would be really valuable to have the actual scans that we've collected and, and identify any biomarkers that come up in those scans across the different clinical groups and how those scans present differently as well. Combining with machine learning paradigms, I think that could really be expanded to a much larger cohort and kind of adapted accordingly as well. Of course, we've got 107 patients. So of course, expanding to a much larger cohort would also allow for a lot more validity as well. And again, this was enrolled just from the Royal Adelaide Hospital. So I think being able to expand even to various hospitals across Australia would add a lot more depth to it. And, ideally combining this with rural GP clinics in rural Australia and collecting those samples as well would really allow for more clinical applications, because the overall aim is to, if someone's presenting with TIA-like symptoms, for them to be able to go to their GP in rural Australia, have a blood test and be able to measure those expression levels and have a response that's fairly accurate and very early on in their clinical process that tells you whether it is a TIA or not a TIA. So then they can be managed appropriately from then on. I've just got my email on there in case there are any further questions, and I'll just acknowledge all the organizations that are involved in the project. And again, I've got a QR code there as well in case anyone is interested in the methodology at all. Thank you.